Hello again, this is Miss Jody coming to you with my second video for the History of Medicine class. And so if you watched my health video, my first video for the health class, you'll know that I am continuing to try to create things for the journals. The journals are really helpful for us because we can use this for review, and of course this is where we keep all of our pictures. With the journals, I will continually send the PDF form of the visuals for you to print out or to create on your own if you don't have a printer. If it's too much detail, like say a drawing like this, and you don't have a printer and you're not going to draw all that out, don't worry about that because this has a lot of detail and all we'd be doing is adding coloring and stuff like that. But if it's something that there's an area where there's lots of good notes to be taken, then you want to try to print that page out. Okay, but I will be printing them out here also and putting them in your journal. So don't feel bad on wasting ink on some that aren't as detailed, only if it's something you have to really draw out in color and put a lot on too, or there's a specific video that goes with each one. All right, so looking back to where we have been in the past with the history of medicine, we obviously have been focusing a lot on the infectious disease side of things and the men and women that contributed to the understanding of medicine that we have today. Now, those are people like Zemoes who said, hey, wash your hands, gross. And we had a nice little experiment with figuring out how gross our hands really were and how we did not do a real good job at washing our hands, which is very important nowadays with the SARS-CoV-2 virus and the new COVID-19 disease. All right, so a while back we went through Louis Pasteur, and he is someone who we're revisiting today with our second time with Louis Pasteur. So as a reminder, Louis is a brilliant scientist, more of a chemist than a doctor, but applying his science to the medical world. And he was one who helped disprove spontaneous generation, along with obviously this experiments Francis Reddy. But understanding that back in the day they thought the microorganisms that cause disease could just spontaneously generate, which was incorrect. Life begets life, and we know that now. And so Louis Pasteur had created this swan neck experiment to show that the microorganisms that would create the wine and products like that to become, what's the word, gross, I'm trying to think of what the word would be, ruining the wine. And so he showed that the microorganisms were actually settling in from the air and that there was a contamination instead of just spontaneously generating. And you remember the swan neck was able to show that oxygen could still get into the broth that he created that did not have any, that was heated so that there was no living microorganisms. And the microorganisms that were trying to come and settle in from the air would get stuck in the swan neck. And he was able to prove that there'd be no contamination because of that. Now, if the swan neck was broke, then they could resettle in and bacteria would be present and it would contaminate the sample. And then also if you had a sample that was pure and then you tilted the neck where the microorganisms could be retouch the solution, then you would have a presence of the bacteria again. And so he was able to prove that these microorganisms did not spontaneously generate. So he worked with the wine, and that was a famous experiment. And then he worked with the silkworms, if you remember how he worked with the diseases that were attacking the silkworms. And so as we continued past Pasteur, we had some time with Joseph Lister. Now, I don't have any notes in my Joseph Lister, and of course, a lot of times I don't, but now I'm putting all my notes in because obviously I'm showing you my pages. And so Joseph Lister, he was the one who helped, and here's some of Shelby's notes here, and have her journal. You know that Semmelweis had really pushed washing your hands, but Joseph Lister said, not only your hands, what about the surgical tools? What about the wraps? What about the air? Everything that you came in contact with, because he was under the understanding, as was Pasteur, that this germ side of things, the microorganisms gone bad, were possibly a pathway to disease. And obviously the germ theory of disease coming from Pasteur, and Joseph Lister being part of this group who believed such. And so with Joseph Lister, we did some experiments where we were trying to create antibacterial solutions with all kinds of wacky stuff to see if we could kill any of our microorganisms that we had grown through our blood agar when we were working on the childbed fever and Semmelweis. And so I don't know how successful we were on that, trying to kill bacteria and also fungi, as you can see there on Shelby's. And so I'll take a little video of our Petri dishes and see what they're looking like, because I don't think we threw those away. I think we kept the blood agar. Some of the other ones were a little iffy, and we might have thrown those away, but I'll check that. So with Lister, the idea was to create something to kill off the microorganisms. Of course, Listerine is named after Joseph Lister because he is that brilliant man who put forth that effort to create a sanitary environment for health care workers. And then there was the search for disease that went along with understanding Robert Koch. And again, I don't have any notes, so I'm just going to pull up Shelby's here. Shelby, yours is getting a nice little view here. And so the search for disease, we started looking at the germ theory of disease, obviously. And Robert Koch was very interested in anthrax. And if you remember, Koch thought that it was very important for us to figure out why was the anthrax coming back. And this led to what's called Koch's postulate, if you remember that in our book. And the postulate just means that it's an idea, it's a way of reasoning, looking at the evidence, like it seems as if one thing leads to another. And so Koch said, okay, in the regular practices, if you had anthrax, if your animals were dying from anthrax, it was very deadly and very troublesome to the normal person that would be raising livestock for their families and for sale. And so what would happen is if your livestock had anthrax, you'd have to burn it, burn the creatures, kill them, burn them, bury them, because all these practices had to be so severe, because otherwise it would come back. And Koch postulated that there was possibly a correlation with some of this. And then eventually they figured out that the bacteria that the anthrax bacteria, which is, I think, Bacillus anthracis, was able to create spores. And any bacteria that can create spores is basically when your condition gets to not be nice and cozy for you anymore, you can create spores so that your type can continue on. And those spores are very sturdy and can withstand circumstances that are not ideal, and then can be reawoken. And so if you let the bacteria remain in the soil, in your habitat for your livestock or anything like that, it would return and kill the next set. So this is why you need extreme care to get rid of the bacteria. And he was able to show that the disease actually did come from the microorganisms, the endospores, and the bacteria, and was able to create an experiment to show this. So he had a lot of contribution to the germ theory of disease. All right, that leads us then into coming back to the time of Pasteur. And Pasteur is someone who has given his whole life to this understanding and furthering his knowledge linking with this germ disease. So if we review, this is just a review page. Now, when I created the journals, I originally created the week we started looking at Louis Pasteur. The title to the chapter, but then I only made one label, which was on rabies originally, because I was thinking, oh, we'll just review a little bit with Pasteur and then do the rabies and then move on. But then I realized that there's so much information with the rabies. With this chapter, that's not just the rabies. There's the chicken cholera and then his fight against anthrax. So I thought, well, maybe I should do a page on each. And I think it's important because it gives us a lot of history, and that's what this class is, History of Medicine. All right, so as a review, and we'll be jumping around because of me having the pages kind of a little all over. As a review, we have microbes that soured wine. He figured that out. Microbes affecting the silkworms. He figured that out. There was a time as he aged that he suffered a stroke, which left him partially paralyzed. He did rally back and eventually learned to walk again with only the help of a cane. And the partial paralysis was an arm and leg. I can't remember if it was right or left, but he did walk with a limp. His wife, Marie, is attributed for being a great support to him. And that is great to see a husband and wife team, and her honoring of her husband, saying things like she tried to always do things and so that he had time for his experiments and so that he could further his investigation. So great to see that in a couple. And then it also talks about how he gave his wife special attention and made sure that he had yearly vacations with her, like 2 weeks at a time or something like that. So after this, in his aging time, and I can't remember how old he was at this time, for some reason 60 years stands out, but I'll have to check on that. He began to tackle a new problem, which was the chicken cholera. The chicken cholera is a disease that affects fowl, so any of the fowl world. And it's similar to the regular cholera, if you've ever studied the regular cholera. And, I don't know if regular cholera is the right term, but it is, let me think, the microorganism is it is Vibrio cholerae, I think, which the Vibrio is a group of microorganisms that are kind of rod-like, but they're like in the shape of a comma, kind of like this shape or whatever, and a little smaller than that. So these Vibrios, terrible, terrible trouble when you get that particular microorganism into your body. And that microorganism for years, and still is a problem in countries who don't have good sanitation, has that still struggle with it, is usually transmitted through the fomite of water. And so water that's not clean, and when you get the cholera, it's like vomiting, diarrhea, weakness. They can't even walk, and it's a really quick onset, really terrible problem. So this particular cholera was cholera that was striking the chickens. Now, it was not the same microorganism, but it is a similar problem. And so what he did, in this, of course, is just the review page. He found the bacteria that caused it. He creates a vaccine of the weakened version. But there's more details, so this is why I decided to jump and create, another page for that. So you would need your own page for this particular one, chicken cholera. So chicken cholera, a highly contagious disease of fowl. It is caused by the microorganism Pasteurella, which is great. It's honoring Louis Pasteur for his discoveries. Pasteurella multocida. I think I'm pronouncing that right, but who knows? You know how I am with my pronunciation. So Pasteurella multocida. Okay, so this microorganism creates the chickens become listless, dropping their heads, same sort of problem with the diarrhea, and eventually die. Always fatal, always a big, huge problem. Pasteur, of course, this was a problem for people who their livelihood was livestock and for caring for their family. And so Pasteur eventually finds the bacteria responsible and begins growing it in chicken broth to study it. And so he spends a lot of time studying this, trying to figure out with the germ theory of disease and what to do, how to stop it. The breakthrough comes when two of his assistants actually give some chickens some of the infected broth for their studies, but it was actually from an old sample accidentally. And kind of like back in the time when penicillin was created, discovered, it was on accident, because in the petri dish the fungi had begun to grow and created a zone of inhibition against the bacteria. So therefore, us realizing you could take one microorganism, a fungi, and use it to fight against another microorganism, a bacteria. And so this was also a accidental discovery. So these assistants accidentally give an old sample. The chickens sicken, but they don't die. And the assistants discover their mistake. They apologize, stating they did not notice the date on the label. But this was actually a good thing, because Louis Pasteur in his brilliant mind is saying, oh, wait a second, then something is with this. The old bacteria gave the chickens a mild case of cholera, and Pasteur wondered if they would now be immune to the more vicious culture that was fresh. And so in an experiment to answer the question, Pasteur injected the same hens with the active culture that they knew was a fresh, very virulent, or a very vicious culture. And to everyone's surprise, the chickens did not get sick at all. And Pasteur, he had known of Edward Jenner's work with cowpox and realized that this was another example of a vaccination. And so even though he knew the word vaccination came from the word cow, he kept the name to honor Jenner. And I thought that was pretty interesting, and shows how much respect they had for each other back then. Now, there is a diagram that I'm going to send you that goes along with this. This one particular goes along with Pasteur's experiments with chicken cholera, and then this one goes along with Pasteur's experiments as he fights the next thing, which is anthrax. Now, I don't think it's important for you to print this out and waste your ink, because there's not a lot of doodads or little areas for notes on this, but you feel free to if you like to cuten these guys up or whatever, but only the ones that have a lot of something I actually want you to write on. That way you don't waste your ink. I will print these out at the Science Center and put them in your books. Okay, so he figures out that if he could use an aged version, which had weakened, of the Pasteur multocida, and injected into the hen, and then the hen's body would create an immune response, and then therefore then that hen was injected with the fresh culture, and there was no disease and no death. But a normal hen of that same fresh culture would be dead. And so with that was the helpful side of protecting the fowl from the actual disease of chicken cholera. So then Louis turns his mindset to the microorganism anthrax. So or the disease caused by a microorganism, I should say, anthrax. So he turns his focus to anthrax. He finds a way to weaken the bacteria and uses it to create a vaccine similar to how he created the vaccine for cholera. But there's more details. Again, this is the information or the introduction page. So I made a page on anthrax, just to understand how the storyline went. So after Pasteur's success fighting chicken cholera, he turns his focus to anthrax. Pasteur read the published reports of Robert Koch, so another correlation between great scientists, Joseph Lister, Robert Koch, all of them working together to solve these hard times back in the day for people who didn't understand this in the health or in the history of medicine. And so he reads the published reports of Robert Koch and his postulate, and he decides to develop a vaccine. Knowing that there was not a weakened version of anthrax to work with, Pasteur decided to try to make one. He said, what if we could make one? And of course, the nowadays versions of making weakened microorganisms, you can use chemicals, heat, aging. So the aging is what they used for the chicken cholera. And so he proposed doing some experiments to see if either chemicals or heat could do it. So after many experiments, success came by controlled heating to just the right point. Because here's the problem. If it was too warm when he heated the microorganism, and of course you have to take the microorganism out, grow it in a substance, something that you can, a solution, something that it would continue to grow in. And so if he heated it too warm, the germs would multiply. If it was too cold, they'd change into their spore form, which again was a problem with anthrax because the spores are so sturdy that they were unstoppable. many times. And so he had to work really hard to get just the right point, and he was able to figure out exactly the right temperature to where it was not too warm to grow or too cold to create a spore, but it was right in the middle to where it would weaken the bacteria. And this bacteria, I think I said already, is Bacillus anthracis, and that is a bacilli, a rod-shaped bacteria. So veterinarians who took care of animals that experienced the deadly pathogen, they were not quick to believe that Louis Pasteur could create this and that it would be successful, of course. But he did create it, and he did experiments, and it was proving to be successful. So he weakened the microorganism, and then he would inject it into an animal, and then the animal's bodies would create an immune response, and then they would then inject the animals with the vicious culture of the microorganism, and they would not die from it. So it was great for them. So the book talks about residual, I can't think of how to say that name, a famous veterinarian, and this is documented because obviously this was in 1882. He had edited an important veterinarian magazine at the time, and he thought Pasteur was basically a fraud and demanded a field test, even after Pasteur was showing a success with his vaccination. So he demanded a field test, and Pasteur was willing to do that. In May 5th of 1882, there was a farm in France that they led 50 sheep into a pen. Half of the sheep were given the shot of the weakened version of the anthrax. So 25 got the weakened version, and the other 25 was the control and had nothing. And then on the last day of May, which would have been May 31st, 1882, so that way it gave time for those who got the weakened version for the body to create an immune response. All 50 of the sheep were then given on that day a powerful, dangerous strain of the Bacillus anthracis, and it was basically a test of time. Would all 50 of them die? And this was a costly experiment because nobody wanted to experiment with sheep because it was pretty expensive to do that. And what if they all did die? Then you'd be 50 sheep out. Louis Pasteur was actually pretty confident, it says, about his experiments because he had done so many, and in the scientific method that if you continue repeat, and you keep getting the same conclusions, you can grow some confidence in your data. And so it did say, though, that he was really nervous as there was a certain amount of days, I forget if it was like 14 days that they, no, that's the rabies, actually, was the 14 days. So I'm not exactly sure how many days, if it was just the next day. That might sound right, that if it was just the next day. But eventually, it wasn't actually that day when they injected, because I know it says in the book that Louis Pasteur was kind of nervous and he couldn't sleep for the amount of time that he had to wait to see if any of the sheep died. So he was confident in his experiments, but then again, you don't know on these unseen things. And so he stated that all 25 vaccinated would survive and all 25 of the control would die in this experiment, and in the end he was correct. And so that famous veterinarian had to give him credit for his discoveries, and this was a big move in the anthrax world, because when it comes to anthrax, it was one of the deadly diseases that they were dealing with at the time, and we don't hear about it a lot in our world anymore, but it's really dangerous. And this little picture is showing them inoculating the sheep and going through that process. So after the anthrax time, then Louis Pasteur was not finished with his studies, and he was continually trying to use this germ theory of disease and this idea of vaccination to figure out some of these deadly and sad things he was experiencing in his day and age. And so as in the introduction from cholera, or the way that he defeated chicken cholera, and then the way he defeated the anthrax, and now he turned his focus to the rabies. Now, we've already studied the rabies, and we did the rabies video when we were here before we ended up getting separated, and but we can review some of that. And rabies was a serious thing. In fact, it still is in a lot of other countries. We don't deal with it as much. We have vaccinations. We have a lot of practices with our animals and domestication of our animals and wild animals are not something that we're constantly in contact with, so we don't have as much, but there's still a lot of rabies problems in our world. And so it was, people feared rabies because it was always fatal. It was not something, as the neurological situation we've seen, how it is slow in its onset, and so you can stop it, but if you don't have the right tools, then you almost always will die. So Pasteur worked with animals who had the disease. He took samples of the foam from their mouth, and he was convinced that the pathogen's path was through the nervous system and due to the symptoms. And his normal plan was normal to find the microorganism, the germ, grow it outside of the body, and then make a weakened version of it, right? And that's how he had dealt with the cholera, the chicken cholera, and the anthrax. But the problem with the rabies is it's actually caused by a virus, and the virus is too small to be seen at this time and understood. And so Pasteur could never find it. And so he can't find it. And then, of course, he can't grow it, because what we know of viruses now is that viruses can't be grown outside of the body regularly, like in a bottle or a batch of soup, like he would do with the bacteria, the broth, because viruses, obviously, they're not alive. And viruses, because they're not alive, they do not have the ability to reproduce on their own. They don't have the mechanisms for that. And so they're just a viral agent. And so you can't make the virus keep reproducing and making more of it unless it's inside a cell. So you needed something living to grow it. Now, nowadays, what we do is we grow viruses in like chicken eggs and stuff like that, so we can create vaccines and such. And so Pasteur said, well, I can't find it, and I can't grow it outside of the body, and so how am I going to develop a weakened version? So what he had to do was he started to have to grow the viruses and transferring it from the foam and such, even though he didn't realize it was a virus, in rabbits. And so if you look at this diagram, this is one that I'm also sending to you. This is just the diagram of the particular virus that for this topic, but this is just a picture of him with growing the viruses in the rabbits and using the rabbit to figure out how to stop. And it's sad because we got bunnies and all that kind of stuff, and it's hard to imagine, but it is what it is, and that's what they had to do back in the day. So what they would do is they would grow the viruses in the rabbits, and then they would sacrifice some of the animals to study what was going on inside. Like, where was just like you'll see when you watch the health video, if you haven't already, in the epidemiology studies of like how the virus works with the COVID-19. Sometimes we have to sacrifice an animal to cut it open and look inside, like where is the virus coming out? Through the mouth, through the diarrhea? I mean, how is it shedding itself? And so they would use the rabbits to figure those things out, poor little guys, and they ended up being able to create a weakened version of the virus by removing the spinal cord of a rabbit that had died of the rabies. And they actually hung the spinal cord by a thread in a clean jar. That's interesting. I'm trying to picture that. And they kept the material hot and dry. And after 14 days, the rabies had lost its strength. So that was their weakened version. So using the spinal cord and the actual rabies being on the spinal cord, and another example of confirmation that this is a neurological situation, that this particular virus attacks the nervous system. So then they realized, well, okay, if we can create a weakened version, but we can't go around just, giving this vaccination to every dog and raccoon and animal in the whole world, what are we going to do? And so I kind of ran out of room here with my notes, and then the next page I had already had here for rabies from the video. And if you remember, like the symptoms and the different type of animals, bats, mammals, dogs, and all that, and the actual family of viruses, the rabid viruses, and the actual genus, the lyssavirus, which means rage, if you remember, and the species rabies. So lyssavirus rabies is the actual virus that causes this. So all of this was something we already did together, and you can use it as a tool to remember why this is such a bad virus, but then also why they're able to stop it instead of injecting the virus in every animal in the world. I mean, the vaccination in every animal in the world, because this is a slow onset, meaning it took a long time, well, depending on where the creature bit you, to get through the pathways into the nervous system, up into the spinal cord and into the brain, that if you could get a weakened version into the body before that virus made its way up to the spinal cord and into the brain, you had a chance. And so as long as somebody was vaccinated early enough before it made its way all the way up to where it needed to start creating the onset of the familiar symptoms, that the foaming at the mouth, what they call the fear of water, the saliva glands, the eyes, the red bloodshot eyes, the just craziness that it creates in you, the rage. If you could get this weakened version into the body so the body could create an immune response, you would be able to stop this virus because it was such a slow onset. And so because I've ran out of room, I put a little of the details down here on this side. So what happened is he started creating this, and people became aware of his success. And so when somebody was bit, it was kind of like, basically, yeah, this hasn't been tested on humans, but my child or my husband or wife or whoever's bit is going to die anyway. And so people just sort of flocked to him for help because there was no other help at the time. So it talks about in July 7th in 1884, there was a young boy, Joseph Meister, and he's famous because he's the first one who gets the actual vaccination after being bitten. And he was actually mauled almost to death, so badly mauled by a mad dog that they thought had the rabies. But Pasteur wasn't ready to use this on humans because it hadn't really been tested, and he was nervous about it, injecting something into a human. But the mom was really persistent, like, he's going to die anyway, and we all know this, so please just do something. So Pasteur gives the permission. He doesn't actually do the injection because he's not a doctor, and so an assistant of his does the vaccination. And this was said to have happened 60 hours after the attack. So this is a few, 224, so 2.5 days afterwards. So the slow onset is the salvation of the time that you have to get this in. And so he ends up being the first person that does not actually die from rabies. And it's such a success story. And I've read a lot about Pasteur, and I remember somewhere reading how he was very terrified of rabies, even from a young age, having seen things, seen people die of it. So it was always in the back of his mind. So to be able to be part of the success story of stopping this terrible disease that was taking the lives of so many, it had to be wonderful. Now, in the end, I don't have any more room to write any more notes, so I just thought I'd summarize this, and that's again because I had my pages mixed up here. But I thought this was really interesting. He does get credit for his discoveries and his work in this area. And then towards the later years of his life, he actually gets honored. And it says on December 27th, 1892, government officials of France and representatives of many foreign countries gathered to honor Louis Pasteur on his 70th birthday. Dr. Joseph Louis Lister was there and had been the first to put Pasteur's germ theory into practice. He attended the celebration, and he represented the Royal Society at that time at the celebration. And Pasteur limped into the hall amid thunderous applause. Joseph Lister stood, walked forward, and shook Pasteur's hand. So I thought that was pretty neat that he got some credit for his life's work, and that many of these other men, Robert Koch, Lister, and all them, who were also passionate, were a part of this. So we'll move on next video to Dr. Linz with the study of scurvy.